based on what we had discussed yesterday, so we are proceeding today. So today we are looking at organic molecules. So what we are discussing were the fundamentals, in that we've talked about the water, we've talked about the bonding and the special properties that water has. So now we are going to talk on about the organic molecules. So, organic molecules, or we could say organic compounds. So, compounds that contain the element carbon are classified as organic and others as inorganic. So, for instance, you would find that carbon dioxide would be called inorganic, but then you have something like methane or carbon tetrahydride, which is um, organic. Okay, so yeah, the human body contains both organic and inorganic substances. That's why you would know that the human body, yeah, has got all these things, carbohydrates, which are organic, and then there'll be ions as well, which we have. We've got calcium, we've got iron, we've got different inorganic things. So this just shows to say the human body contains quite a number of things. So cells contain organic molecules. So carbon has 4 electrons in its outermost shell. So that's something that we had reviewed already. So the fact that carbon has 4 electrons in its outermost shell, and we know that for carbon it obeys the octet rule. So what the octet rule basically means is that carbon requires what? Requires 8 electrons in its outermost shell for it to be what? For it to be stable. So what it will do, it tries and tries and tries to ensure that um, it gets that eight number of electrons in its outermost shell. So this is where bonding comes in. Okay, so this is where bonding comes in. But of course, we know that carbon is more likely to share electrons rather than uh, lose or gain electrons. It's usually more about sharing, and the reasons we discussed them in the previous session. So you can review them there. So uh, these bonds that are going to be formed when carbon shares electrons with other atoms are what we call the covalent bonds, and we had reviewed them, or we've been reviewing them over the past day or so. So, and these are very strong. So you'd have carbon forming bonds with nitrogen in things like proteins, oxygen in things like, yeah, in proteins, in carbohydrates, and then also hydrogen with lipids. So, or any other carbon atoms. So, and this is the beauty about carbon. It's also able to form bonds with itself of varying lengths and shapes. So that's what makes carbon very unique, and that's what makes carbon to be at the center of organic chemistry and organic molecules. Because oxygen, yes, oxygen can bond with itself, but carbon is unique because it's able to bond with other carbon atoms and make carbon chains of varying lengths. So and because of varying lengths, that's why the different shapes come in as well. So that's something that's very important, and so that's why carbon is at the heart of organic chemistry. So long chains containing 50 or more carbon atoms are usually found in living systems. So what does this tell us? It tells us that usually even the things that we would consider to be small organic substances, they usually contain very long chains of what? long chains of carbon atoms. So that's something that's important to appreciate. So we talked about how carbon to carbon bonding can result in what? In different shapes. Okay, so can result in different shapes. So you can have a ring, you can have different shapes, and because of this, it increases the what? It increases the diversity, and because there's an increase in the diversity, yeah, it means that carbon really enables organic chemistry to be a very wide topic because of its many wonderful bonds, especially with itself. So now what happens is that molecules, organic molecules, have what we call a functional group. So what we do is we separate any organic molecule into what we call the backbone. Okay, so I'll explain that in a moment. So small organic molecules such as sugars, amino acids, nucleotides. So nucleotides, those are things which are going to form the nucleic acids, such as DNA or RNA. So these are going to have what we call a carbon backbone. So what a carbon backbone is usually going to consist of is carbon being bonded with what? With hydrogen. So this is where you're going to have carbon being bonded with hydrogen, carbon being bonded with what? Being bonded with hydrogen. So just like that, right? So that's the chain that you're going to have. It can either have a double bond or single bonds, but the key concept is that it's going to be carbon, just yeah, so it's just going to be carbon which is bonded to hydrogen. And then there's usually going to be what we now call the functional group. So the functional group, this can be a hydroxyl or something that you call OH. Okay, so you're going to have OH, okay, the hydroxyl. It can be a carboxyl. So that's the carboxyl is something that you call, that's the COOH, isn't it? The one that is for organic acid. Huh? Then you put the phosphate groups. So the phosphate groups is where you have your chain, then you have PO4 there, right? So this means that now the importance of these functional groups is that they determine so many characteristics of what? Of this of small organic molecules, in that. And why are they stressing that for small organic molecules? Because here is the thing. Uh, let me give an example of an alcohol, right? So you're going to find that certain alcohols, okay, let's start with the smallest alcohol. So the smallest alcohol is methanol, right? Methanol, methanol has got the hydroxyl group. And so because that's a polar group, if methanol is able to dissolve in what? Is able to dissolve in water. Okay? But what happens is that as the backbone chain begins to increase, as the backbone chain increases, as the carbon backbone increases, you see that the effect of the what? The effect of this the effect of the functional group usually in the area of um dissolving can actually be what? Can actually be overcome, can actually be overpowered. And that's why you see that when you get something that's very large, something which has got a very large carbon backbone, and you want to dissolve it in water, it may not dissolve as well. Why? Because relative to the functional group, even though the function group allows it to dissolve, but the carbon backbone chain is so large. And remember, it's non-polar. Okay, so placing it in water which is polar, it fails to what? It fails to dissolve. So that's why here they are saying these functional groups determine the characteristics of what? Small organic molecules. So that's why that small organic molecules is there, right? So that's something that's very important. So one characteristic of an organic molecule of some importance, it is whether it is what? hydrophilic or hydrophobic. So hydrophilic is something that's water loving. Hydrophobic is something that is water hating. So why is this important now? That's very, very important because remember, these reactions or these organic molecules, their reactions are going to be occurring where? Are going to be occurring in water. So what this entails is that knowing their solubility is then important. Yeah. So knowing their solubility is what is very, very important. But remember, just remember what I said to say, even though the function group can allow, so even though the functional group can allow something to dissolve, but also the carbon chain, the length of the carbon chain tends to have a what? Tends to have an effect as well. So that's something to take note of. So now, as we begin to talk about the organic molecules, of course, in human, like we saw there, we said that usually uh, in organisms rather, or in living organisms, what you're going to see is that these molecules are going to be long. Huh? So you're going to find that maybe the carbon atoms which are there are going to be a lot. So now this then introduces us to what? They are going to be in parts, right? So you're going to have a small unit. Then these small units, when they come together, that's when they form a what? They form a large unit, right? So let's now look at that those small components that can then be put together, or which can come together to what? To form the large organic molecules. So an example that I would give for this is, let's talk of amino acids, right? Amino acids are what will form the what? Proteins. Okay. So you talk of glucose. Glucose is what will form starch, isn't it? Okay. So that's the thing, right? So there's going to be those small units which are going to what? To eventually form a large organic molecule. So each of the small organic molecules are already mentioned. So already remember we talked about proteins, lipids, and carbohydrates, right? Okay, so each of the small organic molecules already mentioned can be a unit of what? Of a large organic molecule. Okay, so we've talked about that. And also, with regards to that, it also talks about the organic molecules, the different ones that we've talked about, right? So we'll review them in a short while. So a unit is called a monomer. So a unit, this is part of something big, right? So yes, there's a large organic molecule is there, but there's going to be what? A unit that's going to be there. The same thing as we could have a tray of eggs, but we can say an egg is a what? An egg is a unit. So an egg is a monomer, right? Okay, then when we take now the whole tray of eggs collectively, that becomes a polymer. So simple sugar such as glucose is the monomer of what? Of polysaccharides. So in that, the polysaccharides are going to be made up of what? Of simple sugars. So there we've got the functional groups of some common monomers. So we've got the aldehyde group. So this is just a little bit of organic chemistry, but um, in this is really discussed in more detail in your chemistry, organic chemistry to be specific. But it would be important just to know these functional groups, and more so to know which one is polar and which one is non-polar. So really with this, it's all about that. So the first one, we're talking about the what? We're talking about the aldehyde group. So the aldehyde group, what happens, if you want to recall, is CHO. So what you have is you've got that carbon which is bonded, double bonded to oxygen, and single bonded to what? To hydrogen. Then for the ketone group, that's where you're having the carbon that's part of the function group. It's double bonded to what? To oxygen. So you see, that's more of like CO, isn't it? Yeah. We see that The hydrogen that was on the aldehyde group has been replaced by what? By any alkyl group that can be there. Okay, so there could be an alkyl group, a different alkyl group. So remember, an alkyl group, this is just an alkane where we remove one hydrogen atom. So for instance, we've got methane. So we know methane is carbon tetrahydride. So that's CH4. So when we remove one hydrogen from methane, what do we have? We then have methyl, isn't it? Yeah, and so on and so forth, right? So that's what an alkyl group is. So then we've already talked about alcohol in our introduction. So alcohol, you see there, the functional group is OH, or more specific, or more accurately, you can say the hydroxyl group. So all those places where you are seeing R, it simply means R can be anything, ranging from a methyl group to an ethyl group, just on and on. So that's something important to take note of. Then there is what we call the carbonyl group. Okay, so yeah, so that's about it. But of course, you can see that the carbonyl group, that's something that we saw in the ketone. Yeah, so if you could see, that was in the ketone group, so that was a component. Then you see now the hydroxyl group, or the hydroxide group, or the hydroxide ion, or the hydroxyl group. So basically the same thing. And then you see the sixth one, that's a phosphate group. So I remember there was once a question that we asked, I think I should be in biology, to say how many double bonds does the phosphate group has. So yeah, you may want to review that phosphate group. So you see it's one. Okay, so that's about it. So now you will realize that usually molecules which have a hydroxyl group, or the hydroxide group, or the OH group, are hydrophilic. So why are they hydrophilic? Because when you look at the bond OH for the hydroxyl, we said, remember yesterday we said the electronegativities for oxygen and hydrogen, they're very different from one another. So oxygen is very, very electronegative, and hydrogen is not very much electronegative. So the electrons are not going to be shared equally. So as a result, the oxygen is going to have a slightly negative charge, whereas the hydrogen is going to have a what? Is going to have a slightly positive charge. And so as a result, what you're going to have is that bond is going to be polar. So because it's polar, and we know water is polar, so that substance is what? Will be hydrophilic. But again, remember that reference that I made to the effect of the carbon chain, right? So we don't have to forget on the aspect of that chain. So we've talked about the functional groups in general. So now we're now going to talk about the carbohydrates, to be more specific. So carbohydrates are widely distributed in plants and animals, and it is in here where they fulfill both structural and metabolic roles. So, what does that statement mean? It means that carbohydrates don't just give us energy. So, energy, giving us energy from respiration, that goes under the metabolic roles. But then you've got the structural role. Let's talk about things such as cellulose. Let's talk about cellulose is very, very important in what? In structure building. Okay, in cell structure building. So it's carbohydrates are not only important in metabolic roles, but also structure building. Okay, so you can talk about things such as, yeah, very, very important. So then glucose is synthesized from carbon dioxide and water in plants. So you may be familiar with with the process of respiration. So but yeah, with respiration of, sorry, photosynthesis, right? Yeah, so glucose is synthesized from carbon dioxide and water in plants. So that's a process of for photosynthesis. that reaction is not balanced. Huh? So to balance the equation, you add a 6 in front of the carbon dioxide, a 6 in front of the water, and a 6 in front of the oxygen. So then that reaction is what is balanced. Okay, so glucose is going to be produced in plants, and then uh, that excess which is not used in respiration is going to be stored in what? Is going to be stored in starch. And so that's why we are able to eat maize, because it's just the glucose which has been what? Stored in and what? As starch. Okay, so we'll look at how starch is formed and the types later on. So animals can synthesize some carbohydrates from fats and proteins. Okay, so that's something important to take note of. That's why you would realize that someone that's not eating consistently, they end up losing weight. Why do they lose weight, or why do they appear to be thin? So you find someone was very relatively fat, but then you see that as a result of maybe starving, not eating much, you see that they begin to get thin. What's happening? It's because the fat under the skin is now being taken and used to what? To form carbohydrates. Huh? The same thing for the proteins. So that's something that's very, very important to take note of. Okay, so that's a process you learn about in your biochemistry called gluconeogenesis. Huh? So you are producing glucose from fats and proteins. So that's a very, very important. So that's why you realize that people that are starving themselves, even their muscles are going to be what? Their muscles are going to become very, very weak. Why? Because muscle is just protein, isn't it? So if proteins are being used to form carbohydrates, it then entails that this person is losing muscle mass. So that's why you see that the person will be looking weak and may not really be energetic and all that. Anyway, not really more on the energetic part, but when you see their muscle, you can see that some muscles are what? Some muscles are wasting. So that's something that's what? So that's something that's important to take note of. Okay. So of biomedical importance, so the sugar or glucose is the most important carbohydrate. And why is it? Because the brain needs carbohydrates. Okay, so it's also absorbed into the blood because okay, so it's a major fuel of the fetus of mammals. So what happens again? We know that the fetus gets its energy supplements from what? From the mother. Okay, so to ensure that this occurs, so the blood contains what? Blood contains glucose, right? And that's why for those that may have been familiar maybe with the hospital, and you've seen how people are measured for diabetes, we measure their blood. So we're just simply checking uh, how much glucose their blood has. So there's a particular limit which we say once it increases beyond this, then we're saying okay, this could be diabetes. When it goes, so that yeah, so when it goes above what we expect, that's hyperglycemia, right? So there's an increase in what? In glucose levels in the blood. And when it goes below what we were expecting, that then becomes what? Hypoglycemia. So again, that's something that's very, very important to take note of. Okay, so that's basically about that. So glucose is very important because the brain usually needs what? Glucose. Okay, so that's why we are always worried about amount of glucose that someone has. So then we now talk about the sugars. So solid sugar form crystals and dissolves in water to form a sweet tasting solution. Now, many people usually confuse the term carbohydrates with sugars. So here is one thing. Sugars are one form of carbohydrates. So if you'd want, you'd say all sugars are are carbohydrates, but not all carbohydrates are what are sugars. Okay. So what happens is that a sugar is something which you get the crystals, you dissolve them in water, and what you are getting is a sweet tasting what solution. So for instance, when you get sucrose, okay, you get sucrose, you put it in water, you drink it, it's sweet. You get galactose, you do that, it's sweet. Okay, so these are what? These are sugars, and sugars are into two groups, right? So you can have monosaccharides and disaccharides. Okay, so monosaccharides, of course, mono, so it's just one. Okay, so just one unit. So remember, we are talking about monomers, huh? So it's just one, isn't it? So these things like glucose, fructose, and galactose, isn't it? Then disaccharides, di means two, isn't it? So disaccharides, these are formed from two monosaccharide what molecules, and this reaction is what is a condensation reaction. And what do we know about a condensation reaction is that water is what? Water is going to be eliminated. Okay, so water is going to be eliminated. So don't worry, we'll look at those reactions in detail. So we see how those disaccharides are formed. So glucose and glucose forms maltose. Glucose and fructose or fructose, depending on the pronunciation, that gives us sucrose. Then glucose plus galactose gives us lactose. So you see the one for sucrose and fructose. You see, you can see that it's the only thing. Sucrose and fructose are the only things which have got a U. Huh? Like when we look at on the aspects of galactose doesn't have a U. So that's how you remember to say sucrose comes from glucose and what and fructose. Then lactose and galactose, that's already sorted out, because the disaccharide is just removing the G and A, from the galactose when it adds with what with glucose. So that's just an easy way in which you can want to remember them without really having to memorize. So like I said, all sugars are carbohydrates, but not all carbohydrates are sugars. So sugars are one form of carbohydrates. Okay. So the other is polysaccharides. So meaning what do we have? So we've divided carbohydrates into three classes already. So we've got monosaccharides, disaccharides, and polysaccharides, of which we know of these which ones are sugars, monosaccharides, and disaccharides. So the common question they'll ask is, look, the shatter will just list glucose, galactose, everything, and then put starch and then say which of the following is not a sugar. Then someone will get stuck because they are thinking all carbohydrates are sugars. So the best way to remember this is you don't get maize, maize meal, or what we call the millie meal, right? That's starch. So you don't get starch, and then put it in water and start drinking it, isn't it? Okay, that's why when we are making porridge, after the porridge is ready, what do? We add sugar. Okay, yeah, because starch is a polysaccharide. It's not a sugar, but it's a carbohydrate. So please get these things right and to be very helpful. So carbohydrates are classified as, so these are things that we've already talked about, but we'll just be diving more into them just to appreciate them. So monosaccharides, these are carbohydrates that cannot be hydrolyzed into simpler carbohydrates. Okay, so what this basically means is that when you've got a monosaccharide, so this is a carbohydrate, but when you try to break it down, the moment you try to break it down, then you lose a carbohydrate. Okay, so you're going to have something else, you're going to have another compound, but one thing's for sure is that it's not going to be a what? A carbohydrate anymore. Okay, so you've removed the what? You've removed the integrity. So That's something that's very, very important to take note of. So, having discussed that, so there we see that if you try to subdivide or to divide a monosaccharide, you see, you break it down and you will realize that you can, maybe let's say, let's get glucose for instance, right? So you know that glucose is has got 6 carbon atoms, isn't it? Or in it, right? In its structure. So it's what we call hexose. So we use the number of what? The number of carbon atoms. So if there are 4 carbon atoms, we say tetros. Five, we say pentos. Six, hexos, just like that, right? And that's why, yeah. So when we subdivide, let's say, a hexose sugar, okay, such as glucose. When we break it in half, we are going to have, so we are separating 6 carbons into half. So we are going to have 3 carbon atoms, 3 carbon atoms. So what do we have then? Then we remain with what? With a triose, isn't it? Okay. Then we'll come across the pentose sugars, right? And these are the ones which are very important in these are the ones which are important in the nucleotides. So we have those, and those are very, very important, but we will review them. But the take home point is that when a monosaccharide is divided, is hydrolyzed, it's broken down, it loses its carbohydrate nature. So you can't break it down, into what? Into simpler carbohydrates. So that's the take home point. Then we now have disaccharides. So disaccharides, of course, like we've talked about, these are this is two monosaccharides put together. So meaning, if you hydrolyze, so hydrolysis is just breaking down something, right? Using water, obviously. So what happens in this case is that, remember, and why is water important? Because remember that when a monosaccharide and the disaccharide come together, water is what? Water is eliminated. So for you to restore the to break that bond, you need to bring back water so that those uh, two hydrogen atoms and one oxygen molecule can go back to what? To the monosaccharides, isn't it? So that's why hydrolysis is important. So now what's going to happen is, uh the so you've got a disaccharide. Let's talk, maybe you've got maltose. So when you divide that maltose, when you break it down into half, what you're going to get is what? The constituent monosaccharides, right? So if you were to hydrolyze maltose, you get glucose and glucose. When you hydrolyze sucrose, you get glucose and fructose. When you hydrolyze lactose, you get glucose and galactose, right? So that's about it. Then, yeah, then something that's important is now is to differentiate between the oligosaccharides and polysaccharides, because here many people tend to have a number of so the key thing here is oligosaccharides. These are these are carbohydrates in which, when you break them down, you are going to get between what? Between 2 and including up to 10. So what this basically means is that oligosaccharides, these are carbohydrates which contain more than 2 monosaccharides up to 10. So you can have something that contains 3 monosaccharides, it's called oligosaccharides. Something that contains 7 monosaccharide units, you call it oligosaccharides. Something that contains 10 monosaccharides units, you call it what? Oligosaccharides. Because we know two is what? Two monosaccharides together, it's what? We know that's disaccharide, isn't it? So it's from 3 up to and including 10 monosaccharides units. I hope we've gotten that part. So yeah, so that's something important to take note of there, right? So for the example there, I've got maltose triose. So you see maltose, and then the triose there. So something important to take note of. Then polysaccharides, remember, it's more than 10 molecules of monosaccharides when hydrolyzed. So this means that if you get a polysaccharide and you try to break it down, hydrolyze it, you are going to get more than 10. Remember, if you get 10 monosaccharides units, that's still a what? That's still an oligosaccharide, isn't it? So you have to get more than 10 molecules of monosaccharides when hydrolyzed. So that's something that's very, very important. And of course, an example of polysaccharides is starch. So that's something important to take note of. So then we want to talk about monosaccharides, the fact that they contain only carbon, hydrogen, and oxygen in the ratio. So this ratio is very, very important because it actually helps us, to have an idea of really what our our molecular formula is going to be. So if you want, you can look at it like something more or less like an empirical formula, right? So yeah, so monosaccharides contain only carbon, hydrogen, and oxygen in the ratio of 1 carbon to 2 hydrogen to 1 oxygen, isn't it? So you have all those ratios. So so the empirical formula is that CH2O and then brackets N. So you know N is what? N is the number of carbon atoms. So how then do you know what number of N to use? Remember I said you can have a triose, you can have a pentose, you can have a hexose, right? So hexose, like we've been talking about, has got how many carbon atoms? 6. So you make n what? N to be 6. Because remember, when you multiply through n, your carbon is going to have what? Your carbon is going to have 6, isn't it? So you're going to have 6, 12, and so you're going to have carbon 6, H12, and oxygen 6, right? So that's the empirical formula, and that's how you can derive the molecular formula for any monosaccharide. So they would ask you to say what is the molecular formula for a monosaccharide, or they would ask you what's the empirical formula for a monosaccharide. So you should be able to differentiate them as we've done there. So yeah, so for today we end there. So we'll continue looking at monosaccharides and then also continuing there. So I don't know if there are any questions with regards to what has been done.